Well, kia ora tātou everyone, and welcome to the webinar. We're just right on time at the moment, and the people are still joining. It just takes a little time for the technology to allow everyone to join in. So we'll just give a minute or two, and then we'll get underway. But it's wonderful to have you here at this webinar today. Well, we like to try and stick to the golden hour, so we'll get underway. And of course, it's recorded, and so folks can come back and listen to the recording if they want to. And of course, the session has been repeated at 7 o'clock this evening, so you're welcome to return this evening. But it's my great pleasure to introduce Associate Professor Louise Parr-Brownlie to you this morning. Louise is from the University of Otago. She's an associate professor. Just reading off there, because I think like many of us, we find our knowledge of the brain is not very good. So we can't do it off the top of our heads. But it says that Louise is internationally recognized for her expertise in understanding how activity in the motor thalamus, motor cortex, and basal ganglia control normal movements and is altered in Parkinson's disease. And so the topic that Louise put down was about this thing called brain inflammation. So Louise, we're really looking forward to you unpacking this into layman's terms to us parkies who don't quite understand what's going on. And we're just so grateful that there's people like you in the world who spend your lives working all this very complicated stuff out for us. So thank you, and over to you. Thank you so much.
Tēnā rā tātou he mihini nui tēnei kia koutou. So hello and welcome. I want to say thank you for choosing to spend a little bit of your day with me, because I'm going to be sharing a bit of a story about how my research has changed and moved into being very much interested in understanding how neuroinflammation may be able to be modulated in order to slow down progression of Parkinson's disease. So I'm just going to share my slides with you. Just need to put that into presenter mode. Okay. Are you able to see just the one single slide at the moment? No, you're seeing me.
You just in a small corner, and we can see your slide as well.
Okay, that's great. Wonderful. Thank you for confirming that. So my first slides. Part of what I'm going to be doing today is sharing a little bit of a story around, I have a role as the director of 81 National Science Challenge, so I very much think about aging, very much think about Parkinson's disease because of my personal research background, and I think about equitable outcomes, and that is why I've ended up changing the research direction that I'm actually moving in towards. So I'll be explaining my research background. I'll be talking about the aging trajectory and weaving in how things change with Parkinson's and that as well. I will look at the inflammatory system. It will be very light, and because it's not the focus that I want to spend the time on, I really want to explain how we might be able to harness it in order to slow progression of Parkinson's. The link between the gut and the brain is much stronger and tighter than we ever believed. So I'm going to start explaining that and look at some modifiable risk factors for Parkinson's that we may be able to harness in order to reduce inflammation. And reducing inflammation at any point in the lifespan, but actually throughout the whole lifespan, is probably what we are advocating for. So my background, I came to the University of Otago as a physical education graduate or undergrad, and I was very much focused on health and well-being. I came in because I really like sport, and I wanted to know how we might harness all that knowledge for performance in that way. But of course, once you're here, you learn so much more. And I started to be very much interested in how the brain controlled movement and how changes can occur in a condition like Parkinson's disease. So as I went into my postgraduate study, I started to record from single brain cells, and I was using a rat model in order to be able to collect the data. So I've got some data here to just show you how we have traditionally done experiments in my lab. So we are looking on the y-axis here at 80 cells that we've recorded, and we're looking at their average activity over a 1 second period. And so each horizontal line, it has a series of dots to represent the activity for that one cell. Time 0 here was when our rat was reaching for food. So we know that the rat was moving, but the movement was actually very good in this situation because we are recording from a control rat. What we find is that during certain periods, the firing rate of the cell changes. So around time 0, which is when the rat's paw obtains the food, there was an increase. So this reddy yellow color indicates that the firing rate increases, but periods either side of it, just before and after, the colors are very blue. And that indicates that the firing rate goes down. So the key thing to get from that is that firing rate of cells and across many cells within the brain are critical for underlying a really good, functional, well-executed movement. Of course, we conducted experiments in a Parkinsonian rat model as well. And what we found is that, surprisingly, when the rats did actually execute movements, we did see this increase in activity, and I did not expect to see that. What was unusual is that the decreases in activity were altered. So what we know is there are specific changes in activity that underlie the movement deficit. So these movements were slower, and in fact, these Parkinsonian rats really didn't move much at all during the experiment. And that these changes are critical for underlying the movement deficits. So I record that information because actually I want to look at how we might treat Parkinson's differently and of course find eventually better treatments. So one of the things that we've been spending a lot of time on for the last 12 or 13 years is a technology called optogenetic stimulation. Essentially, this is really similar to deep brain stimulation, which people within New Zealand do have as a treatment option and is offered to some people at least. And deep brain stimulation is incredibly effective for reducing dyskinesia related due to the levodopa, and also for improving movements very generally. However, some people that receive deep brain stimulation, when the stimulation is on, people can experience speech changes, gait disturbances, obsessive thoughts, and depression. So there are some side effects. And so the whole purpose of looking at this optogenetic stimulation technology is that we were hoping that we would be able to develop a technology that has fewer side effects. And the reason for that is that the technology is incredibly specific. And I'm going to explain how we do that in just a moment. So this is one of our rats on study. It is a Parkinsonian rat, and it is meant to be reaching into a feeding tube just here. And just below this red arrow is a little Cocoa Pop, so a wee treat for it. This rat has been trained to actually reach for the Cocoa Pops, but of course it's Parkinsonian, and he's exhibiting akinesia, so he's not really moving at all. He knows the food's there. He could move anywhere in that box, but he's still orientated towards the feeding tube. He just isn't executing the movements. So with optogenetic stimulation, what we were doing is that we take a viral vector that is going to contain the genes in order to express a protein. That protein, we want it to go into specific brain cells, and when it's there, a flash of light is going to open that protein and the cell will become active. So what we can essentially do is control brain activity in some specific cells in the brain. So what we've got here is this rat. It's the same rat that you saw before on the same day. It's just, when I start the video, it's been undergoing 20 minutes of blue light stimulation. And the blue light, the flashes of blue light, is what's going to open that protein and cause the cells to be active and hopefully recover the movement. So what you will see if you watch really carefully in this region just here is that he does reach for the food and he is able to grab it and he consumes it. So if I just play that. So we found that with certain patterns of stimulation, we could recover function. And subsequently, since then, a rat brain is small. We've been changing the technologies because blue light doesn't actually transfer very far in the brain, in a brain. And so in a human brain, if this is ever to go to patients, we'll need to use a different color of light. So we've been changing the protein, changing the color of light stimulation so that eventually it might be a treatment option. And we've had really, really good success. But what also happened, about 5 or 6 years ago, I took some time and I was just stopping and thinking about what I was doing and how I was doing it. And we just received a big grant that we have completed that work on the optogenic stimulation. But I stopped and thought, you know what? We're not actually slowing progression of Parkinson's disease. We're not preventing Parkinson's disease. Am I really doing the right thing? And in addition to that, the people who are offered deep brain stimulation, they are often younger people. One of the criterion around whether you'll be selected is your age. So if you are older, over 70 years of age, it's often not going to be offered. And so there is an inequity around deep brain stimulation. And I didn't feel comfortable about that. And from my own whānau, if we're the brain, the head is a restricted region, putting in a viral vector, putting in something that's actually going to be sitting and stimulating the brain is not something that very many members of my family would find appealing. And so there may be some cultural or ethnicity groups that are going to be disadvantaged as well. So at that point, I really stepped back and thought, you know, maybe we should look at some more holistic and health and well-being approaches, and maybe we'll find some different ways of approaching all of this. And I was really lucky as I was making those decisions. Elodie Kipp joined my lab. She's actually from Belgium. She has a background in virology and inflammation. And so we started having conversations, and eventually, along with some other colleagues, we have been very much redirecting the research program that my lab will be doing in the future. So to share some of that journey. So this slide shows the aging trajectory. So from at the left-hand side, from the moment of conception, right through to a much older age. And so the reality is we age from the moment of conception. But what does happen is we can age differently. So people generally talk about chronological age. What we know from the Dunedin multidisciplinary study is that they took their participants at 45 years of age and looked at their biological age at age 45. What they found was that some people, their biological age was much closer to 55 and others were much closer to 35. So our biological aging can vary a lot. And that's determined by a number of environmental factors that can be modified. And of course, the actual chronological age is a major factor. We can't change the number of years that we have actually lived. So there are a number of factors that affect how well we age. And we wondered whether we could harness some of that knowledge and think about new ways of potentially treating or slowing the progression of Parkinson's disease. So I'm first going to talk about the genetics of Parkinson's because this is relevant. It's not modifiable, but actually there are environmental factors that do play into this. So less than 10% of people with Parkinson's disease have a genetic cause. We know about 5 or 6 years ago that there are 15 causative genes. And so I've got 2 examples, PINK1 and SNCA is the gene for alpha-synuclein, which is a protein I'm going to talk about very soon. And these causative genes significantly increase the risk factor for getting Parkinson's disease, and are often associated with early onset Parkinson's disease. For the majority of people, as I'm sure you all know, that get Parkinson's disease, they have an idiopathic onset. They tend to have the disease diagnosed around 60 or 65 or older, and there's no known cause. What we know here is that there are 25 risk genes. That does not mean the person is going to get Parkinson's disease, but they do have a greater risk. And then on top of that, all those environmental factors that are important for aging and the aging trajectory add to that. And so the environmental factors can actually exacerbate the risk for Parkinson's disease. But if we use them well, maybe we can reduce the risk of Parkinson's disease expression, the onset of timing of the diagnosis. So what we've been very much doing is looking at harnessing the knowledge to reduce the Parkinson's disease risk. And our overall goal is to slow symptom progression. Before I go on and talk about the specifics around the details around what happens when we age and is associated with Parkinson's disease as well, I really want to pause. Our society, Western societies as a whole, are obsessed with young and youth. And not everything about being young and is great. And there are a lot of good things that come with aging, such as wisdom, experience, maramatanga. Generally, older people have a healthier outlook. We know what's important. We know what's important to us. And they're the things we spend our time on, and don't worry about that other stuff. And if people are at that point where they're no longer in paid employment and they're able to get superannuation, then they have time to do the things that they want to do. What I've found out is most people are still incredibly busy. They're just doing the things that they really, really love. So there are good things about aging. And I know for me, looking back, I don't want to be 15 or 25 or probably not even 35. So I'm hoping that when I get to 65 and 75, I still feel that same way. So changes with aging. So as we age, there are changes that just naturally happen, and we can't alter that. So the first one, so regeneration and repair is reduced. So we know that if we have a cut, when we're really young, it might only take a couple of days and it's really well healed. But as we age, it might take 4 days or 5 days or even longer. As we age, the amount of muscle mass that we have reduces. It means we're not as strong. It means that our posture changes. The change in posture means that we can be prone to fall injuries. And because we have reduced bone mass as well, if we fall, then we are more likely to have a fracture. There are sensory system changes, and our sensory systems become less effective. So I'd like to blame my eyesight on getting older, but unfortunately I ended up having to wear glasses at 10 years of age. So, but hearing, vision and balance do tend to deteriorate as we age. For Parkinson's disease, one of the symptoms that often comes on early before the diagnosis is that there is a reduced sense of smell, reduced taste as well. We're aware that as we age, metabolism slows. We can't eat the same amount of food that we used to eat when we're about 20. But actually there are really specific changes that are occurring. So when we look at the metabolism and we look at the brain the glucose is usually the energy source used by brain cells for over 80% of the time. And what happens in Parkinson's and other neurological disorders is that those brain cells can't use the glucose the same way anymore. We need to find another fuel source. And that's important because by taking more glucose on, it actually isn't going to solve what we're trying to address here. It's actually just going to predispose us to diabetes. Gut dysfunction alters. So swallowing, absorption of food is reduced. And one of the early symptoms of Parkinson's disease or before its diagnosis is often constipation. altered, the gut function alters. What we also know is that the bacteria that's in the gut is very much altered, and I'll be talking about how that has informed experiments and might be a future treatment. As we go into the brain repair is impaired as we age. So there's proteins need to be broken down. So there are proteins that we don't want to accumulate. They need to be broken down. You basically have a Pac-Man kind of system that gobbles them up and breaks down those proteins so they don't accumulate. The function of that breakdown is actually impaired as well. Normally, one brain cell will connect with thousands and thousands of other brain cells, and it's not just one connection, one cell to one, it's one cell goes to that other cell hundreds to thousands of times, and it's doing that to thousands of other cells. So we get a reduction in the number of connections. Now, some of that pruning is fine. We do have, more than what we really need, but there becomes a point where it will alter function as well. And as we age, more brain cells die. We do have the ability to regenerate or create new brain cells, but it is quite slow, and they do have to track to all the different parts of the brain. There are only a couple of sites that the new cells are born. And so, yeah, the regeneration is going to be incredibly slow. For Parkinson's disease, of course, the cells that die off are going to be dopamine cells in the midbrain, and one of the reasons they do that is because the protein alpha-synuclein accumulates there. And then finally, as we age, the immune system is slower at fighting pathogens, and what that means is that we get a chronic low-grade inflammation, which is sometimes called inflammation. And I'm going to bring all of those elements that are in bold there together to explain what we think might be important as a future treatment. The place we're going to start teasing things out a bit more is actually in the gut. So in this diagram here at the top, this yellow region, this is the inside of our gut, the small or large intestine tube. And this is where our food is. There will also be bacteria that is there as well. As we get towards the gut wall, which is these green epithelial layer cells, there are going to be mucus layers, and the mucus layer traps things that we may not want to get into our body, and it's our first line of defense. But our really critical line of defense is these epithelial cells tightly lined up, which very much restrict what goes across. So we want our nutrients across, but we don't want bacteria and other pathogens to actually get into our body. So, because this is a site of high risk of pathogens getting in, there's a lot of the immune system is well represented here. It's going to be everywhere, but it is well represented here. And I'm just going to very briefly talk about the immune system. There are two parts to it. The innate part of it, this is the part that goes directly to the pathogen, say this bacterial molecule here. And these cells, a macrophage essentially acts like a Pac-Man, engulfs the bacteria, degrades it, and spits out the content. So it neutralizes it from getting further in our body. So the macrophage has an important role. When we get to the brain, it's called a microglia, and also astrocytes have an important function there. So these microglia, or macrophages, and other cells that are dying off will release cytokines and chemokines. These are chemical signals that tell us that something is not right, that we've got a pathogen in our body, and it's going to attract other parts of the immune system to that site so that we can contain it and stop ourselves getting sick. So the key thing around these cytokines and these chemokines and how the macrophages, microglia and astrocytes work is that depending on their state, they can increase inflammation, or we can harness them to reduce inflammation. There are also other cells that are important, natural killer cells, and there's a dendritic cell, but we're really not going to talk about them at all. Another part of the immune system is the adaptive immune system. So this is the system that when we get immunized, say against COVID, this is a system that works. It has the memory system so that the first time we're exposed, the T cells see the foreign pathogen come in, it engulfs it, it presents what we call the antigen. It presents a part of that cell, part of that pathogen to the outside, so our immune system knows that it needs to respond, and then the B cells start to produce the antibodies, which is really targeted to one specific pathogen itself. So this adaptive system has a memory. So once you've been immunized, then the next time you're exposed to that pathogen, then we'll recruit that adaptive system really quickly, and hopefully we don't ever get sick. So the key thing about these immune responses is that they need to be recruited at the right time, and they need to be balanced, and sometimes they get out of balance. So the gut has that lining of epithelial cells. In this figure here, it's called intestinal cells, those that are really critical for creating a barrier. But actually, sometimes that barrier becomes leaky. And this slide explains some of the factors that can cause our gut to become leaky. So food, in particular, food that has high in saturated fat, very much processed food, high in with a lot of sugars, and also stress. These are two factors that are possibly under our control. And if we have excessive levels of not so great food choices or high levels of stress, then it can predispose for our gut to become leaky. In addition, toxins, bacteria, and of course some medications. So if you take antibiotics, what it will do is it will target and break down the bacteria in our body that are causing us to become unwell. But we do have bacteria along our gut lining as well. And what it tends to do is reduce the number of good bacteria, and therefore the bad bacteria actually flourish. And that's why we often end up not feeling so well from those medications, or end up with diarrhea. But the key thing here is that the gut can become leaky, which means that these products, these pathogens, can get into the tissues just underneath, the muscle layers. It can get into our blood, and then those pathogens are going everywhere around our body, and we end up with a heightened immune system, or we've got systemic inflammation. And if this keeps happening again and again and again, what sometimes happens, or if the immune system is overactive, we can end up with autoimmunity disorders, and we can end up with food tolerances. Now, the gut and the brain are incredibly well linked. So when something happens down here in the gut, the brain does know about it. So the contents that are taken across the gut will end up in the blood. We just know that the immune system can be really important and can be triggered if required, and the immune cells will end up, or the pathogens will end up going around the body and trigger immune responses everywhere. There are endocrine links between the gut system. So when we've eaten, hormones are produced. Those hormones are going to go up to the brain. They tell us that we're full. There can also be hormones that are really important for telling us we need to eat. We're actually hungry. We've always known that there are nerve links between the brain and the gut. So when we have the parasympathetic system, that generally means that our gut becomes more active. So when we rest and digest, we relax. Hopefully that's when we're eating. The breakdown of the food happens really effectively. But actually, we know that these nerves can transport proteins, and the protein that's going to be really important in Parkinson's disease is alpha-synuclein. So this diagram here is showing you a situation where the gut is really leaky. We have a lot of bacteria, these little green spots here that have come through, and it's setting off the immune response. And as a consequence of that, within our gut wall, within the muscle layers just underneath it, the cells are no longer able to get rid of the alpha-synuclein. So we do produce it normally, but we don't let it accumulate. But it starts to accumulate because of the inflammation down at the gut. That alpha-synuclein then can travel up the vagus nerve to the brain, and at that point, the alpha-synuclein can accumulate at the pons or the brain stem. The other way that the alpha-synuclein can end up accumulating in the brain is that the pathogens can enter through our nose. It gets to the first bit of the brain, the olfactory bulb, the tightness between what enters there and can enter the brain is not very tight at all. So once the pathogens are in there, if they're accumulating, then again, we can get alpha-synuclein accumulating in brain cells around the olfactory bulb. And then no matter where the alpha-synuclein is accumulating, it will generally cause cells to die off and the alpha-synuclein spreads. And so it can spread to other parts of the brain. So we have alpha-synuclein accumulating up in the brain that causes neurodegeneration, and it's all happening because there's actually neuroinflammation as well. So just to put in a little bit more context that I was talking about before, here's our leaky gut. Here is the pathogens getting across and getting into the blood. When it does that, it affects the nervous system that's really close, just actually in the gut wall. It sets up an inflammatory state. We then end up with systemic inflammation. The blood-brain barrier, so that barrier I'm going to show in just a moment, is really, really tight. It then gets leaky, and then we end up with neuroinflammation. And so we've got a whole series of events that are predisposing to changes in the brain and cells dying. So what we know is that there are gut symptoms that could be potential biomarkers. So the fact that many people with Parkinson's disease, before they're diagnosed, look back and go, oh yeah, I have been constipated for years. There are things like the gut microbiota, the bacteria in the gut have changed, the diversity is much smaller, and the good bacteria are no longer there, and there's going to be gut inflammation. Now, knowing this is really useful because it means that it can help direct us onto some treatment options. So the first treatment option is going to be diet, and I am going to be talking about some diet options very soon. Another treatment option, if the bacteria within your gut has changed with Parkinson's disease, what they have found is that they can do fecal transplants. So they've taken fecal matter from someone with Parkinson's disease, they've put it into a mouse, and that has been enough to cause that mouse to actually get Parkinson's disease or to express the alpha-synuclein. What they've also done is take fecal matter from someone who is really well. They've transplanted that into someone with Parkinson's disease, and it has improved motor symptoms for at least 6 months. So although this is not routinely offered, it is a potential treatment solution. Another one is that the vagus nerve. So I just talked about the alpha-synuclein going up the vagus nerve. It can also go down. So one treatment is actually they cut the vagus nerve very close to the stomach at the truncal root. And when they did that in people that they knew had a high risk of getting Parkinson's disease, they found that it reduced the risk for over 20 years. So they had to do a full cut at the truncal site, because if they only did a partial one, then actually there was no protection. So when we get up to the brain, and I talked about a leaky brain, what we've got here is a blood vessel, and inside is going to be our red blood cells and our white blood cells, our immune system defense. So whatever is in our circulatory system, for that to get across into the brain, the barrier there is incredibly tight. It lets very little across. So it does happen to let T cells across, but it doesn't allow other cells such as B cells to go across. So what happens when we get a leaky brain? This is complex and we're not going to go into it, but the balance here between, here's a microglia. This microglia is in its resting state. it can be activated and become so that it's fighting the immune system, so it becomes pro-inflammatory. When it does that, it produces a lot of these chemicals, the cytokines, and when that happens, it can actually increase the degeneration of neurons in the brain cells, and in particular for Parkinson's disease, it's going to be dopamine cells. So inflammation in the brain is not a good thing. But we can, if we're able to harness this inflammatory system, then we can actually force these microglia into an anti-inflammatory state. And then the cytokines that it releases are actually good ones and are going to reduce the number of cells that die off. So we can harness the system. So when you have a leaky gut, we have a leaky brain. When we have a leaky brain, we've got neuroinflammation that's occurring in there. So there is evidence of neuroinflammation in people with a range of neurological and psychiatric disorders. So what they do is use positive emission tomography. Basically, they use a radioactive label and they can decide what they want to label. So, for example, they can label dopamine receptors to see what the level of dopamine is in the brain. But in this case, they've labeled microglia because they want to see if inflammation is occurring. So in Parkinson's disease, what we know is that microglia accumulate in that brainstem area, the pons, and also within the basal ganglia, the parts of the brain that we know that are affected in Parkinson's disease. In Alzheimer's, similarly, the cortex is affected and the hippocampus. So for all of these disorders, the microglia accumulate in the sites where we know the pathology changes. So there's definitely neuroinflammation occurring. So I started right at the earlier talking about the aging trajectory and that we may be able to harness some of that knowledge in order to how we age and possibly harness that to slow progression of Parkinson's disease. So really our body, the example here, the analogy is the tree, the leaves are the outer part of our body, but what's really important is the roots in this case are what's happening inside, and that's really hidden. So we want to understand what our body is experiencing and therefore how we might be able to harness that to actually reduce inflammation in the long run. So what we believe is that reducing inflammation over the lifetime is a positive thing to do for aging and for Parkinson's disease. And what it's going to do, what we'll be trying to do, is maintain a healthy gut-brain axis. So hopefully down the bottom here are a number of ways that, or a number of things that it can increase inflammation, or on the left-hand side, decrease inflammation. But as a general rule, the sorts of things we want to do to reduce inflammation are exercise regularly. And this is walking. This is walking stairs if you're able to. This is not marathon running or some crazy level of exercise here, just exercising regularly. We want to eat quality food, so unprocessed, lots of fruits and vegetables, a diet that's low in saturated fats, high in omega-3, as much as possible, because if you have less financial resources, then often you don't have the choices the same way. We need to care for our brain. So here we want to reduce the risk of traumatic brain injury, so concussion. And if someone does get concussed, then we need them to actually rest so that they can recover fully. And under this caring for the brain part, I've also put in here that as we age, we may need to look for a non-glucose energy source. Sleep is incredibly important, and in Western societies is undervalued. It's like, oh, I'll not sleep so much, because I've got all these really important things to do. Actually, the rest and recovery is important during sleep, and clearing of proteins and things that have been broken down in the brain only occur at the deep stages of sleep. And we know that because we discovered a new lymphatic system, which is called the glymphatic system, and that specifically takes away those accumulated proteins that we don't need there. And of course, we want to maintain balance. We need to reduce stress. Our lives are far too stressful. We need to stay connected, not on devices. This is talking to people, having experiences, sharing, being reminded of what we love, and here, just enjoying being with people. And the last one I've got there is spending time in nature. So we know that by spending time in nature, our mental health actually improves. But I saw something come out the last few days that in a particular type of forest, there were enzymes that were around that actually reduced people's neuroinflammation as well. So there's more going on there than we've previously realized. So how might we do this? I'm going to talk about some dietary interventions that we've been exploring. And in fact, there are some good data about how they might be useful for Parkinson's disease. So this study, this table here, what this study tells us about is they had a group of people with Parkinson's who had a low-fat diet. They had a second group of people with Parkinson's disease who had a diet that had a lot of fats, a lot of quality protein, and was low in carbohydrate and glucose. They wanted to see if when the people took this ketogenic diet, this drives our brain to use a different source, which is called a ketone. And if we do that, what was the outcome for people with Parkinson's disease? What they found is that on the ketogenic diet for 8 weeks, and this was actually done in Waikato in New Zealand, they found that Parkinson's disease motor scores improved and the people's quality of life scores, this is on the Parkinson's disease rating scale, in case you're aware of it, they also improved. Interestingly, they repeated the study with people living with Alzheimer's disease, and they found similar outcomes, positive outcomes because of this diet. And the way that this diet works drives the brain to use a different energy source, and then as a result of that, it reduces inflammation in the body. The second example here is intermittent fasting. This is when people restrict their eating to about 6 or 8 hours within the day, usually during the daytime. And that means they've got a long, prolonged period where they're not eating or they're fasting. And what we've found here is that the metabolic effects are very similar to the ketogenic diet, and ultimately the inflammation in the brain is reducing. And then the last one there is the Mediterranean diet. Diets high in polyunsaturated fats or monounsaturated fats are also incredibly good because they reduce neuroinflammation. So one thing that might be able to be used, harnessed by anyone on this video is that you could consider one of these diets. So preventing and reducing inflammation is important. There is a balance. So we can either reduce the things that increase our risk for Parkinson's disease. So that's things like stress, exposure to toxins, looking after our brain if we've had a traumatic brain injury. But we can also increase protective factors. So that's your exercise and having a healthy diet. There are 3 things here that I haven't mentioned yet. So nicotine, caffeine, and alcohol in moderate levels, as far as Parkinson's disease go, actually reduce the risk. The way that this seems to be working is when these are consumed, it changes the inflammation in the gut and it resets it. And therefore, I'm also assuming that they help reduce neuroinflammation as well. So having moderate levels of alcohol and caffeine, I would actually recommend. The nicotine, I don't, and that's purely because the effects on the respiratory system, the cardiovascular system, the other effects are going to outweigh and not help people's health and well-being at all. So what we've been doing is applying that knowledge to what changes in the brain with metabolism, with aging, and with Parkinson's disease. We've been thinking about, well, what is the best diet? What evidence is there that diet actually can alter neuroinflammation? And I've just shared some of it. But what we really want to do is harness the benefits without actually having to have the diet. So think back to New Year's and people have their resolutions and they kind of go, oh, next year I'm not going to do whatever it is. And so often these resolutions fail. And that's because behavior modification is incredibly difficult. So knowing that, what if we can take the metabolic effects that occur with, say, the ketogenic diet and we apply those, can we have the benefits as well? So we're doing the experiments to explore that. We've started those experiments to explore that at least. But in the future, what I'd be hoping is that clinical trials will show that by modulating inflammation, there'll be positive effects for Parkinson's symptoms, that there are already a range of immune system modulators and anti-inflammatory drugs that could be used. But I do put in a note of caution. Their use is incredibly complicated because of that whole balance. It's such a complex system. So if you are going to at least explore in that direction, definitely seek medical advice. So what we're advocating for is that we need to reduce inflammation throughout our whole lifespan. And by doing that, the reason we know it's important is that by reducing your inflammation, it reduces the impacts on pathology that is going to be causing Parkinson's disease. We know that balance is key, and balance throughout a whole of our life will be important, and that by harnessing this knowledge, we're hoping that we may be able to prevent Parkinson's disease in some people that have those risk genes. If we can reduce the environmental factors that add on to that, we might be able to prevent them ever getting Parkinson's disease or delay the onset. And of course, we're hoping that reducing your inflammation will slow symptom progression. One of the things that's really that drove me to actually move in this direction is that I'm advocating for treatments that are equitable. Everyone can have them, regardless of their age or their ethnicity. And we are testing metabolic targeted dietary interventions. And just lastly, I'd like to acknowledge there's a lot of people on the slide, so I'm not going to go through them. So on the right-hand side, there's the brain stimulation research that I introduced right at the beginning, and the people or the funding agencies that supported that research. And on the left-hand side, some members of my lab and also the collaborators that I've been having the discussions with around neuroinflammation and how we might harness that. So I'm going to stop sharing my slides and welcome questions.
Thank you, Kira, Louise, thank you so much for that. Very interesting, and we'll just wait to see if anyone's got any questions that they're going to bring in. But for myself personally, really interesting, because I before I was diagnosed in 2019, I had about 3 years where I had terrible stomach issues. And at the time I was working for Fred Hollows, and I was traveling right around the Pacific, and I saw a number of specialists who basically said, well, you must have eaten something. There must be something in your gut that's got you upset. The moment I was diagnosed and went on to Matapar, whilst it hasn't cleared up completely, I'm significantly better than I was previously. I am on a restricted diet, but it feels to me like, I've heard other people say that we've got as many, the gut has basically got as many neuroreceptors as the brain. So there's this connection, it seems, between the two. Have I understood that correctly?
Yeah, so within the gut wall, there is the enteric nervous system, and the enteric nervous system can operate independently so that you get what we call gut motility, your gut contracting so that the contents get forced through the system. But in addition to that, we have the nerves that come down from the brain that can actually make that work stronger, the contraction stronger, and force the contacts through either faster or slower. So they are really intimately related, and important.
Right. So one of the things that I did, and one of the questions that's come up, is that I tried a variety of probiotics. I spent a lot of money on the ones that are in the fridge and the ones that are not in the fridge. So one of the questions that's come up is, are probiotics helpful? Is it a good idea for us to take them?
So probiotics, I have never tried them, and I haven't specifically looked into whether there've been any clinical studies around their use and whether they're effective for either reducing leaky gut or for Parkinson's symptoms overall. What I would be wanting to know is probiotic needs to get into the small intestine in order to be able to do its action. And one of the things you've got to go through is the stomach, which has a lot of gastric acid. And so it needs to be surrounded in something that protects it at that stage, or else it will denature those probiotics in order for them to get down. So, but if we stand back and just go, you know, gut wall, the gut lining does have bacteria. Some of them are good and some of them are bad. And if you're putting in some good bacteria, then it may well have positive outcomes. And at the end of the day, you are your best, you know, you can do your own experiments in that regard. If you find that they're useful, then you can keep using them. And if you do find them useful, of course, the other thing you can do is stop using them for a little while and go, oh, does everything regress? And then you know that it is actually worth spending the money or not.
Right. So one of the other questions is, you mentioned a number of diets. So, you know, some things are palatable and some things aren't. So can we mix it up a bit and, you know, choose bits from this diet and bits from that diet? Or do we have to commit to one particular type of diet and that's it?
That's a really good question. I think if you take... So one of the things about the ketogenic diet is incredibly difficult for most people to adhere to the level that we're supposed to do it if you use it. So you can reduce and do it, you know, not quite strongly, not quite as strong, but there will be a level that you've actually got to achieve. So mixing them up. And so if you're thinking about, if I do some fasting and actually have some ketogenic parts, it's probably a good combination because you are doing things that kind of complement each other, rather than if you're trying to do something where you've got a Mediterranean diet, which usually does have quite a lot of carbohydrate in it, and trying to do ketogenic diet, then they're not going to mix very well at all.
Right. And you know, we're such a dairy producing country. Is there any research on, you know, that we could drink too much milk, or that we should go into the soys and the almond milks instead, or anything in the dairy area?
Not that I'm aware of. So when I've been doing all the reading around neuroinflammation and Parkinson's disease and the gut, I've not come across that, but then I haven't specifically looked at that as a question either.
So someone saying that they're, you know, really excited by the research that you've been talking about, and asking, could you tell us kind of like what's the future look like? Where are you headed next?
So what our experiments that we're doing, we are testing, there are a range of products that are available now that we believe would be really useful for reducing neuroinflammation. There is some evidence that they can reduce symptoms of Parkinson's disease already, and there are clinical trials that are nearly at the end where we're waiting to see those results. And one of them is vitamin B3, niacin. And so it's an essential vitamin, absolutely, but actually its function in the gut and what it does in inflammation is very specific, and so it might actually have its own little action. And so we've got about 4 or 5 products that we think are really could be beneficial. Of course, what we believe is that combining at least a few of them together is going to have even better effects than having only one of them alone. Wow. So that's what we're doing our experiments on.
So this question is that the person has heard that mannitol, which is an artificial sweetener, can help decrease the alpha-synuclein, and also have heard had information about red light therapy. Are you able to comment on either of those?
So mannitol, I don't know too much about. Never seen around that. Red light therapy. So red light. So when I was talking about optogenetics, and I said, oh, we need to take it to the human brain, which is much larger. We actually were using red light for our most recent experiments. But what we do there is actually put a fiber optic probe. So our phone lines, specialized bit of that, into the brain of our rats, and we splash the red light and we can activate our cells. Red light actually can get through, and far red light can get through the scalp and the skull. But of course, as you're trying to get deeper and deeper into the brain, the transmission gets reduced. So there is some evidence about red light being used for a range of treatments. Again, I haven't looked at it as a general source because we've been very much focused in on the optogenetics, which actually you need to have that protein that you specifically express that's not normally in our body.
So one of the other questions is, any advice when parkies take antibiotics, what we should do?
Follow the instructions around whether you eat it with food or without food, because of course, if you're able to take them with food, then it's the effect that it's having in the gut is going to not be as great. And I guess at that stage, if you're taking antibiotics, look at those dietary components. What can you do that's going to reduce inflammation? And whether that is around considering some fasting periods, whether that is around following some elements of a ketogenic diet. The thing around the ketogenic diet, though, is you can't just do it for one day and it's going to work. It has to be several days in a row and it has to be ongoing. So it's a little bit harder to go in and out of doing that. Taking on healthy elements of diets, yeah, for sure. Exercise, that reduces inflammation.
Is there a problem with weight loss, let's say dramatic weight loss, because you've changed your diet and you're exercising more? Is there any danger there?
So for many people, when they're looking at a ketogenic diet or fasting, the reason they're doing that is to target weight loss. So that is a concern for people with Parkinson's disease. In that study that was done in New Zealand, 8 weeks, they found that there were no changes in blood lipids, or no significant changes in blood lipids. They also found that the weight did decrease a little, but not a lot. It's something that if you're going to try it, you definitely want to keep an eye on your weight as well, because most people with Parkinson's disease cannot afford to lose weight.
Wow. So a question asking you, you're talking about the importance of sleep. So let's say someone's had a career in working shift work and had disrupted sleep. Could that have been a cause of their Parkinson's? A
cause? Probably too strong a word. It might have contributed to it. So if you think back to that idea about there are some risk genes and there are a number of environmental factors, it might have been the sleep disruption might be one of the environmental factors that did actually change a person from being at risk to actually ending up having Parkinson's.
And then, I was also very interested when you spoke about the fecal transplant work. So someone's asking, is this available in New Zealand? And if not, any sign that it might be?
I actually haven't talked. No, I'm not sure. So the work that I am aware of was done overseas. I'm not aware in New Zealand of whether it's been available or not.
So I'm just going to read this one off. When will the optogenetic stimulation going to be used? I've been diagnosed in 2005 and wanted to try DBS. I'm 75 years old with chronic pain problem.
So the question is, when will optogenetics be available?
Yes.
Yeah, okay. So
who's more out 75.
Yeah, so where things are at with the optogenetics, it has been trialled in people, but around vision, changing vision function, and it has been shown to be beneficial there. What I was contributing to, first of all, the evidence that we can modulate brain activity and recover function, and then around the technologies, one of the things I was working with some bioengineers, because in order to do optogenetic stimulation in people, we need to have a device that's implantable. So there is a device available for animal experiments, but at the moment that hasn't been upgraded, if you like, to humans that I'm aware of, and that's a critical step. The research around the viral vectors and whether they're safe, I think there's evidence that that's okay. So it's actually the device is where the limitation is at the moment.
Right, so some more quick fire ones on diet. Turmeric, have you?
Definitely.
And is there a minimum amount of fasting that is beneficial? Or well, what's the minimum that would actually have an impact, I suppose?
Most people use something like a 16-hour fast, 8 hours eating or less time eating. So I think from what I've seen in the literature, it seems to be 8 hours. Other people do approach the fasting a slightly different way. They use it as a resetting of the brain. And so they'll do it periodically. So whether that's every 3 months or once a year. And in that situation, someone might fast for a week or so. So for someone who is Muslim and goes through Ramadan, where they're fasting for at least a week, then that can help reset the state of neuroinflammation or inflammation in the body, and might be useful. So there are different ways you could approach this. You can go for a shorter, but like you do it constantly, or you can go quite hard. You do it for a week or a week and a half, and then you might only repeat that every few months.
So this is an interesting one. So is there any evidence that gut inflammation disturbs sleep, and so you get into a bit of a cycle?
I don't think I've read specifically that link, but from what I've presented, I wouldn't be surprised.
Right.
Yeah, because the gut disturbance is going to create neuroinflammation, and therefore a lot of the same things are going to apply.
B1 therapy, any knowledge about that?
Is this vitamin B?
The question doesn't say that, but I would presume so.
So B1, not that I can remember. I did mention the niacin, which is vitamin B3.
And then I can't pronounce, I don't think, triglyceride oil, or glyceride.
So a particular type of oil or fat, triglycerides. I think from memory, I think you want to reduce triglycerides.
Reduce.
Yeah.
Right. So we're out of time, but the questions are still coming in. So thank you to everyone who's been asking the questions. It really does make it very interesting at the end to hear what you've been asking. Let me just see if I can just find one more for us to. If I've listened to you today and I've got a whole lot of questions around what am I going to do about this now? I feel I'm in a kind of like a dietary chaos. Do I go see a dietitian? Do I go to my GP? What do I do?
So I think start talking to your clinical support people. So yeah, your GP is definitely a good person to talk to. Neurologist, if you can get access to a dietitian, that can be really helpful. I am aware that so I was talking to someone who had young onset Parkinson's disease a few months ago, and they've always been a strong advocate for being aware of their diet and controlling their diet. And they asked to see a dietitian when they saw their neurologist, and yeah, they were really not being referred at all. So there seems to be like using something, because this is really about holistic and well-being. So stepping back and using these approaches by at least some in the clinical sphere tends to be kind of like, no. And yet, diet's really fundamental. So why wouldn't they? Why shouldn't you check your diet?
Just to remind folks that our Parkinson's educators have got up-to-date information on things like diet, and also have the capacity to refer you into another service. So just contact Parkinson's New Zealand. If you haven't signed up as a client, you need to go through a referral process, but it's very simple. Just on our website, download the referral form and send it into the email address, and one of our Parkinson's educators will be in touch with you and will be able to help you navigate the health system if that's what is required for you. And also, just before I thank Louise, just to make a plug that our next webinar, our final one for the year, is on the 14th of December. There will be more information coming out, but it's with Rachel Howell. And Rachel is a physiotherapist who has done special qualifications in movement. So a little bit different to the normal exercise, so movement. And Rachel, we're going to be doing it to Christmas music. So it's a Christmas special on the 14th around us Parkies maybe getting some boogie on. And what can we do, which is just right at the right time for Christmas to get that on. But to end off today, Louise, thank you so much. Wow, you covered a huge amount of ground. It obviously is incredibly technical, but you managed to make it that we could grasp hold of what you were talking about. And so we just want to say thank you, thank you, thank you. And thank you for being willing for us to record this, because some questions are saying, is this going to be available? Anyone who's registered for the webinar, you will be sent a link in about 5 days' time. It takes us to upload the seminar, the webinar rather, to our YouTube channel. And so you'll be able to then watch it at your leisure as well. And the slides will also be available. I think the slides are going to be available. Louise, are you releasing this?
I can see them through, yeah.
Yeah, so those will come out for you as well. So thank you everyone for your participation. It's been lovely. There is a repeat of this at 7 o'clock tonight if you'd like to come and listen again. And you just need to sign up. It's a different link. You just need to sign up again for the 7 o'clock one. But Louise, thank you so much again. We really appreciate your time and your expertise. And I look forward to seeing you tonight.
Excellent. Enjoy your day.
Thanks, everyone. Take care.